Hello everyone, I am Martina from Imagina Biotechnology, and today I want to tell you that we are launching our new kit. It's the nano-tRNAseq Pro, an upgraded version of our 6 barcode kit, but now it has 12 barcodes and it's gonna be even more powerful to use. So we have now a few questions that researchers may be interested in, and we will try to answer. So the first one is, why does tRNA biology matter and why has it been so hard to measure at scale? Short answer will be that tRNAs are central molecule to protein synthesis, but they are remarkably diverse. Indeed, we have around 400 isodecoders, 40 isoacceptor families, and up to 11 post-transcriptional modifications per molecule, all of which are biologically relevant. The problem is that until now there was no scalable way to measure them properly. Indeed, classical methods like real-time qPCR or microarrays only give you a partial picture, and there's simply no commercial kit available that provides at single molecule resolution both abundance and modification status for tRNAs at the same time. We solved this by combining a custom library preparation that was specifically designed to overcome the structural challenges that typically break standard sequencing workflows with nanopore sequencing and a dedicated bioinformatics pipeline. The result is the first integrated workflow that gives you the full tRNA landscape in one single experiment. The next question is, but what can we actually measure with this nano-tRNAseq Pro kit that we couldn't before? So with the nano-tRNAseq Pro kit, you can see in one single experiment all the mature tRNAs that are present in your sample. This means every CAA-tailed translation-ready molecule. So their relative abundance, but at the same time also their post-transcriptional modification status. And this last part is what makes it really unique. And because nanopore sequencing reads native RNA directly, any modification that alters the current signal gets captured exactly as it is, without the need of any indirect proxies. So in one experiment, you are now finally able to get the full picture. Then another question would be, but why does 12 sample multiplexing change how you design experiment? And working in the lab, I can say that multiplexing 12 samples on a single flow cell fundamentally changes your experimental design, because basically it cuts sequencing costs. And at the same time, it improves data robustness, because more samples mean having better statistical power, but without the need of multiplying your runs. Compared to our previous 6 barcode version, you now have twice the flexibility, because, for example, you can run all 12 samples on a PromethION in one shot, or you can decide to split them across 2 MinIONs runs of 6 + 6 samples, whatever fits your experimental needs better. Additionally, we have also optimized barcode recognition and ligation efficiency, so now the multiplexing is even cleaner. Importantly, I need to add that the workflow remains identical to the one that you already know, so it's not a new protocol that you need to learn from scratch, it's simply a better kit. Again, who is this kit for? What questions is it suited to answer? Well, we can say that this kit is for any researcher who is working on translation, but also epitranscriptomic and gene expression regulation. It is designed to answer questions like, how does tRNA expression shift across different conditions? But also, what happens to tRNA biology when you test different drugs? What happens to the enzyme that modify them? Or again, what happens to tRNA modification when you knock out an enzyme that is, for example, a writer or an eraser? Additionally, you can also look at tRNA charging. And when you combine it with our ribulase technology, you can directly compare the tRNA pool used by ribosomes against the total cellular pool, which is where it gets really powerful. Then the last question would be, how does it compare to other tRNA sequencing approaches? We can say that compared to other tRNA sequencing approaches, nano-tRNAseq kit is the only single molecule method on the market. You can sequence one entire native molecule at a time with no need of reverse transcription nor PCR amplification. That means that you will not have retrotranscription biases nor PCR duplicates, and in this way you can reduce the multi-mapping issues that typically affect short-read approaches. And because it's a direct RNA sequencing approach, the modification signal is captured naturally without any additional chemistry. On the cost side, it's actually more affordable per sample than Illumina-based methods, where you need to sequence significantly deeper to get comparable coverage. So thank you for listening to me, and if you have any other questions or curiosities about our product, please visit us at Imagina. Bye!